Yeah, so hi everyone, I'm Conor Stanley. I'm the principal biomedical scientist in the flow cytometry lab at Guy's and St. Thomas's Hospital, working for Synnovis. And this is just going to be a brief introduction to flow cytometry and how we use it in our lab. So, next slide. Yeah, so these are the just a brief agenda. What is flow cytometry? We'll talk about the immunophenotyping of leukemia and lymphoma, what we do in specialty hematology, and then at the end of the talk, I will go through some example plots from patient cases. So here are just a few examples of when to use flow cytometry. So in the diagnosis of acute leukemias, chronic leukemias, plasma cell dyscrasias, you can use it in lymphomas, so non-Hodgkin's and Hodgkin's. Immune monitoring, for example, the use of rituximab therapy, paroxysmal nocturnal hemoglobinuria, myelodysplasia, and then also we can use it for measurement of residual disease. And this is just a little diagram of how samples are processed for flow. So first, we get our sample from the clinicians. We had a peripheral blood or bone marrow, or we also do fine needle aspirates or CSF samples. We stain the samples, which means we add the antibodies, which are tagged with fluorescent tags. We add that, we stain the cells, we then lyse the red cells to get rid of all that noise. Then we acquire on the flow cytometers, and after that we do analysis using specialist analysis software. So what is flow cytometry? It's a technique for detecting and counting suspended particles or cells and chemical structures when they're in a fluid as they pass through a beam of laser light, and one at a time. So one at a time is very important. We want to catch the cells one at a time, single file as they run through the flow cell. It allows us to analyze simultaneously the physical and chemical properties of a cell or particle. And ultimately, it just shows us what's going on inside and on the surface of a cell. So the flow cytometer itself is split up into three main systems. We have a fluidic system, which comprises of a light source, which is generally lasers. So we have 3 lasers in our analyzers at Guy's and St. Thomas's, and those lasers hit the sample in this flow cell. The light that's scattered by the laser hitting the cell is picked up by these filters and detectors. That's actually the optic system. And then the signal is picked up by this optic system, converted into a digital signal, which is then read out on the computer screen as this series of dots. And the way to think of it really is that each one of these little dots is a cell. So yeah, the fluidic system itself is we analyze cells that are suspended in an isotonic fluid. We pass those through a flow cell, and we apply pressure to this stream of sample as it comes through our flow cell, and we squeeze it so that we have one cell passing through at a time, and the laser can hit that one cell. And this is just a little animation. Hopefully it works. Yeah. So now your sample should come through, and then we shine a laser at it. Cells come through, and you can see that the light is scattered. So after the fluidic system, you have the optic system. The optic system looks very complicated here. The optic system is where laser light of a single wavelength is directed at each cell at the interrogation point. So we call this point here the interrogation point. That's where the laser hits the cell. Light is scattered in various different directions. So there's a detector which is in line with the laser beam, and that's what picks up what we call forward scatter. And then there's also a laser perpendicular, which we call side scatter. And I'll explain what those are in a minute, the significance of those. So at the interrogation point, the laser light also excites any fluorescent chemicals bound to the cell. So as well as this basic scatter of light in this direction and that direction, we also get a scatter of light, which is the fluorochromes that are on the cell surface that we've stained, lighting up when the laser hits them. So the fluorescent light emitted at different frequencies dependent on the color. This passes through filters and is detected by specific detectors. And the signal then is amplified by, in the past, it was photomultiplier tubes. Now it is avalanche diodes. So yeah, this is just a little description of the electronic system. The PMTs, or the avalanche diodes, as they're now called, for each detector converts the light signal that's picked up to an electrical pulse, which is then amplified and recorded. Those digital signals can be displayed on the PC, and we use specialist software to manipulate that data. And we can actually arrange it into these dot plots, as you see here, or we can have histograms as well. And each one of these clusters, so each dot is a cell, but each cluster will show us what population is present. Yeah, so what is side scatter and forward scatter? So side scatter, or forward scatter, sorry, is essentially when the light hits the cell and then the detector behind that picks up a scatter of light, and it'll tell us how big the cell is. Generally tells us how the size of the cell. Separate from that, we have side scatter, which is an indication of the granularity of the cell, or how complex the internal structure of the cell is. So we can actually get a lot of information from just those two parameters, just forward scatter and side scatter. We can generally tell most of the populations within the sample. So here we've got a dot plot which has side scatter on the Y and forward scatter on the X. And as we said before, the side scatter is a measure of the internal complexity in the cell. So cells that we, like white cells that we would expect to have quite complex internal, internal complexity would be the granulocytes. So yeah, you can see here that they're sitting quite high up on the side scatter on the Y. Next in line, you have monocytes, which is again this little cluster here. And then down here where you have no granules present and not very much internal complexity, you have lymphocytes. And then next to that, you have red cells and debris. The larger the cell, the higher the forward scatter. So here you see these cells are quite large, and lymphocytes aren't as large. And then the greater the amount of internal structures, the higher the side scatter. So as we explained, the granulocytes have quite a lot of internal structures, so they are sitting quite high up on that side scatter. So then, as well as forward scatter and side scatter, we have fluorescence emission. So the light hits the cell, and on the surface of the cell are these antibodies, which we have bound to antigens on the surface, and attached to those antibodies are these little fluorescent tags. Those fluorescent tags then emit light, which is picked up by the detectors within the analyzer. There's a little animation. The setup process, so we take blood, we create a suspension, we add antibodies to the blood, we then incubate in the dark. After incubation in the dark, we lyse the red cells, and then we wash away those red cells and the debris, and we run it through the analyzer, and we pick up the data. So this is another animation, just sticking to the surface. Laser shines a light, and then that light is picked up by detectors within the analyzer. So we've had some very good recent advances with flow cytometry. In the past, you had to set up multiple tubes with multiple antibodies added to each tube. So as you can imagine, this was quite time consuming and led to a lot of errors. As you know, if you're sitting here aliquoting antibodies in and someone walks in or the phone rings or something, you look up for a second, you immediately forget where you were and you don't know if you've added the antibodies. It's happened multiple times to everyone who's worked in the flow lab. Nowadays, we have this new technology called duroclone technology, which is dry down antibodies. So we don't have to do this aliquoting out of liquid antibodies anymore. We simply pick a tube off a shelf, we add the sample to it, we incubate, we lyse, and then we run on the analyzer. And it has removed quite a lot of heartache. Oh, happy day. So we have, there are some technical complexities with flow. So one of the issues that you can come up against is a thing called spectral overlap. It's essentially where each detector should only pick up light from the fluorochrome that's emitting in that wavelength. But what can happen is you get this spillover. So you can see it here. You can see it here spilling over into this detector as well. And what can happen with that is that it's difficult to know where the signal is truly coming from. It's a bit like when you have paint here and you're mixing paint. It's hard for us to know how much of this green signal is coming from the blue or the yellow. So what we have to do is we have to try and find a way to resolve these two peaks so that we get a separation and we can only pick up what's truly fluorochrome A and only pick up what's truly fluorochrome B. And in order to do that, we use a process called compensation. And compensation is essentially we run single tubes with the fluorochrome that we want. We run a single color of each fluorochrome, and we tell the analyzer where the positive and negative populations are. And we essentially say if a signal comes within these parameters, that is all we want you to focus on. Outside of that, we want you to delete it. And we build up what's called a compensation matrix. And when we run a multicolor panel, so if we run 5, 6 colors, the analyzer knows that a signal picked up outside of the ranges that we've set can be ignored or removed. And it helps us essentially avoid any false positives or false negatives. So for instance, here with these populations, you can see that this FITC here is, the compensation is very poor. And in order to resolve that, we can create this compensation matrix, add it to the analysis, and it pulls it back in line or reduces the compensation. So now we've got a true population here, which is PE positive, but not FITC positive as it was appearing initially without the compensation. Go back there. Similar down here, again, this population is showing a streak up into this PE region. And if you weren't au fait with what compensation is, then you might think, oh, this population is showing positive for PE. But in fact, once you compensate, it pulls it back down in line, and it shows us that these populations are both PE negative. So how are we identifying the cells? So we use antibodies against specific lineages of cells or cell types. And we use combinations of those markers to tell us what's happening with those particular cells. So some examples would be CD13 and CD33 for myeloid cells. Lymphoid cells, for B cells, you have CD19 and CD20. T cells are generally CD2, 3, 4, 5, 7, and 8. Plasma cells, CD38. Immature cells or blast cells are generally 34 positive and CD117 positive. Platelets are 41 and 61, and then red cells would be generally CD235A. So a really important plot for us in the flow lab is the CD45 versus side scatter plot. And CD45 is an antigen found on all white cells, and it's not on red cells or platelets. So it's a pan-leukocyte marker. And generally, the expression level of CD45, so how bright it is or how far to the right on the x-axis it is an indication of how mature the cells are. So if you have quite weak, as we call it, CD45, or if a population is falling down in this area over here, then we generally think that those could be immature cells. So from the CD45 against the side scatter, again, similar to the way we used the forward scatter versus side scatter, we can get a lot of information from just these markers. So just side scatter and 45. You can see here, bright 45, or yeah, quite high up on the x-axis, the CD45 population. These are your lymphocytes. Next up, and remember the side scatter was an indication of how the internal complexity of the cell looked. So just slightly higher up on that, we have monocytes. And then again, as with the plot we showed a while ago, the granulocytes show quite a lot of internal complexity, and they're high on the side scatter. Down here is what we call the blast zone. This is where, as I said, generally you find immature cells. So we'll have what we call a gate here. These structures that are drawn here are called gates. Essentially, they're just a way of us to, a way that we can focus on certain populations and get the data from each area of the plot. Yeah, so this is very useful in determining populations in immunophenotyping. Gives us quite a lot of information. So we have a diverse testing portfolio at Guy's. We do chronic immunophenotyping for lymphoid neoplasms, acute immunophenotyping for diseases such as AML and ALL. We run a Cesare panel for cutaneous T-cell lymphomas. We do CD19 and rituximab monitoring. We run platelet glycoprotein tests, and we also do lymphocyte subsets. This looks quite scary, but it's just an example of what we run for each of our acute tests that come in. So if a patient has a query acute leukemia, we'll run this 4-tube panel that gives us a broad amount of information on what type of AML or ALL the patient could have. This is our chronic panel, again, broad, allows us to pick up quite a lot of NHL or non-Hodgkin lymphomas, and it's very good for determining CLLs and then also T-cell issues with the tube 1. And this is an example of the FAB system, and I'm just using this because we have a few examples coming up. So here's a quick case study of an acute panel that was run in the lab last year. So it was,
Conor, sorry to interrupt you. You might want to go back and just go through that FAB system for everyone, just so that they know. Thank you.
Yeah, no worries. So this is a way of determining what type of AML patients have, and it's based on the maturation and the immunophenotype that we see expressed on the surface of the myeloid cells or the blast cells. So here you have M0, which is minimal differentiation. You then have M1, which is AML without maturation. So there's a slight difference between the two. M2 is with maturation. So generally we see an M2 coming through the lab, M2 and M4. M3 is promyelocytic leukemia. So keep this in mind because I'll be showing a case of this in a minute, where we have 1333 positive, HLA-DR negative, and CD34 negative. M4 is myelomonocytic. M5 is monocytic leukemia, where the AML is more aligned with cells from monocytic lineage. M6 is acute erythroid leukemia, so more erythroid or red cell related. And then you have acute megakaryocytic leukemia, which is more aligned with, megakaryoblast or platelets.
So I'm just going to add. So you haven't visited Zambia, so you're you don't fully aware of this. But for the Zambian team, I know that a lot of you are actually looking you have the morphology available only. So what Conor's done is related the morphology, and then he's put with them what you might expect to see in those classifications if you had your flow cytometry with them. Obviously, in the UK, we're using the morphology, but we're working with the WHO guidelines more, where we will have molecular characterization and flow cytometry as well. Now, and I know that that's very difficult for you in Zambia, that you have to send them away overseas and pay per cell marker, etc. But Conor's, and you would be more used to seeing the morphology, but Conor's going to go through some flow cases for you now. Thanks, Conor, just to give them background.
Absolutely. Yeah, so this is a case study, patient that came in last year, 43-year-old female, and the clinical details just said new leukemia. So we set up our acute panel, which, as I showed you a while ago, was that 4-tube panel, which had a broad array of markers in it that were focused on both myeloid disease and on lymphoid disease. And our tube 1 here, reminder that we use our forward scatter versus side scatter to pick out our populations and to pick out our live cells, or to separate any debris as opposed to actual cell information. Then we use our side scatter against CD45. And remember, I said that we could pull out a lot of information from that. So here we have our lymphoid cells, our monocytes, our granulocytes, and then falling in that region that I had mentioned about the CD45 weaker population is a big population. About 94% of the cells that we ran were blasts. Then further on from that, you can see that that red population, the blast population, is showing up as it has CD117 expression, it's CD34 negative, and what else is there? CD33 positive. So we would be inclined to think CD33 positive, it's probably a myeloid disease. 117 positive as well, it's probably myeloid. Now, we're seeing here HLA-DR negative and CD34 negative. And if you remember from the FAB classification that we showed a while ago, HLA-DR negative, CD34 negative, 117 pos, and CD33 pos showed up as promyelocytic or APML. So we'll continue to look through. There's no 13. And that might be important. There. The MPO is positive, which is very important. And then, yes. So this one was interesting, actually. I think we go back to this. So initially, we considered APML, promyelocytic leukemia. However, the 13 was negative in this patient. And the morphology also didn't quite fit. It didn't quite fit with this, cupping of the nucleolus, and it just didn't fit with the morphology that we were seeing. We were seeing, more cupping. And what actually turned out to be was an AML with an NPM1 mutation. And what can happen is you have the same phenotype. So from flow, it's not always perfect. You sometimes do need to run these molecular markers. In this case, the patient was considered to have an APML, and the patient was treated with ATRA. But a molecular myeloid gene panel showed that the patient had an NPM1 mutation, which was quite good, actually, because NPM1 is a very robust MRD target. So the patient can be treated, and then in future we can just look for that mutation and see if the patient still has measurable disease. So the next one is a 21-year-old male who came to the hospital, and the clinical details were given as query acute leukemia. Now, we ran it again with the acute panel, which again has the array of myeloid and lymphoid markers. And in this case, again, huge population of the CD45, I would say negative, in this case, CD45 negative. So there's no CD45 expression. And the 93% of the cells run were blasts. Now that population, the red population, is showing up as 34 positive. It's also showing up as 117 negative. My immediate thought would be that this isn't myeloid. There's no 117, but also a key giveaway is that it's all 19 positive. It has very little or some CD20 expression, which could indicate immaturity, and then also it is TDT positive. And there's something going through the chat. So then the final diagnosis for this patient was B acute lymphoblastic leukemia. So 34 positive, 117 negative, 19 positive, and DR positive. So these three markers here really giving it away: 19 pos, DR pos, and 10 pos. The CD20 was positive and negative heterogeneous. There was also cytoplasmic CD34 positive and TDT expression. So BALL. This is an interesting one. 67-year-old female, had a lymphocyte count of 58, hypercalcemia was 3, and then also B symptoms and a known IgG MGUS. That was the clinical details that we were given. On this patient, the CD45 expression on the population of interest is actually quite bright. So we would think maybe it's not an immature disease. Maybe there's something more kind of mature here. We picked it up. Site scatter 45. We picked out this population, which was 80% lymphocytes. And then we saw that that population was all CD3 weak. The population was all 4. So it's 4 CD3. And then in our second tube of that chronic panel, this population was all 19 negative. So not T cells, or sorry, not B cells. These were all T cells. And 20 neg. And then in this tube, this population was all CD7 neg. It was all 25 pos. So 3 pos, 25 pos, and then a drop off or no CD7 expression. And we would normally expect to see CD7 expression on T cells. And it turned out that this patient had adult T cell leukemia lymphoma, or ATLL. 3 pos dim, 4 pos, 8 neg, 2 pos, 5 pos. 7 egg, and 25 pause. And the morphology with these flower cells. And then the last case was a 49-year-old male, new diagnosis leukemia. And this is similar to the one that we saw a while ago again. On the y-axis here, this is mislabeled. This is from where I worked before. This should be CD34. So it was, again, sitting in this blast area or those CD45 weak area. CD45 weak, CD34 positive, TDT positive, and CD19 positive. And again, just hammering home that that is BALL. And that is everything from me. And now happy to take any questions.
Does anyone have any questions? I think probably.
Good afternoon.
Hi.
Thank you for the presentation. It is quite simple, but the problem that like it's a good start for our team here in Zambia. Although like many of us doesn't have a knowledge about the flow cytometry. So I think this is encouraging for us to start reading about it and knowing more about it. And thank you. I feel it is quite simple. Thank you.
Yeah, relatively, yeah, it is. And it's and you can get quite a lot of information from quite a quick test. So that's one of the real benefits of flow, is how fast the test is and how much information you can get in such a relatively short amount of time. And it's generally the first test that clinicians will look to, especially in cases of hematological malignancies. You know, you could be waiting weeks sometimes for molecular results, whereas if you're quick about it, you could have a flow result in under 4 or 5 hours.
But you have to make sure that you have it set up right, and
Yes.
you know how to analyze the data. Otherwise,
Correct.
you can make some mistakes.
Yeah, the whole process is only as good. The data that you get out of it is only as good as the care that you put into it.
I know Catherine and Josh are on the call. Catherine and Josh, did you have any comments? Josh has got his hands up, I can see.
Yeah, thanks. That's a really good talk. So thank you very much for that. My morphology always got a lot better when I knew what the results of the flow cytometry were.
Correct.
In fact, the older I got, the more I relied on the flow cytometry to tell me what I should be looking down the microscope. And I don't think I'm alone in that, though maybe people still are very proud of their morphology skills. But the number of times that I got it wrong without, so it's just to emphasize the power of the technique. So it's a bit of a question. Obviously, to go from not having availability of flow cytometry in Zambia and to where would you start? Because going straight to the sort of full package of flow cytometry would, I imagine, be very costly and also very, very skilled.
Yeah.
Very, very skilled team you'd need, and that wouldn't happen overnight. So where would you start if you wanted to start this in Zambia? What would you use?
It depends on, I think it probably depends on what you're looking to analyze for, or what testing you're looking to do. I mean, I know that there are very good packages. So for instance, Beckman Coulter have an analyzer called the Aquios, which is kind of a plug and play analyzer, but you're limited in the panels that you can run on it. But if, let's say, you wanted to just look at lymphocyte subsets, then it's a really good solution. It is essentially just plug and play. You load the sample on, and it spits out a result for you. You then have to be able to read the result, but it is quite simple to set up. In terms of setting up the other panels, I think, like, I don't want to sound like a salesperson for Beckman Coulter, but they do have, they have now developed these Duraclone tubes that have all the antibodies built in, and you can just buy off the shelf. So rather than having to, because some of the real difficulty in flow cytometry is designing and working up your panel from scratch, whereas with these clear labs, they're called clear lab tubes, it's kind of all done for you beforehand. So you set up your analyzer. That is a difficult part. You have to set up your analyzer, but you can just buy in these tubes and run them. But I don't know, the expense could be prohibitive.
Yeah, I was going to say, they sound like more expensive options. And my general experience with plug and play machines is that you know, it's very easy to just think, I'll get the answer out. But there, as with any machine, as with any bit of kit, there are always problems. There are
definitely
things that don't, a bit like your APML that wasn't an APML, that sort of thing, which you can make a misdiagnosis on with the plug and play things if you don't fully understand the systems.
Correct. Yeah. If I mean, start small. I think you start small.
Sorry to interrupt you, Conor. I was just going to say, I think that the freeze down dried antibodies are quite a good thing for any lab, in that the storage conditions are less onerous and they have a very long shelf life, Josh. So that's quite a good thing.
Minimum of 2 years, I think. Yeah. So I've spoken to someone from Beckman Coulter recently who said that they pulled one of these down off a shelf that had been there for 7 years. Officially, they validated them for 2 years, but they ran it and it looked perfect. So, I mean, that's anecdotal, but they really don't seem to have that much degradation over time.
Unlike liquid.
Unlike liquid, exactly.
I got a quick question for our colleagues in Zambia, actually. I know you send a lot of stuff away. Well, not a lot of stuff. You know, you send some samples away, but my experience of delays in delivery of and poor handling of samples sometimes means that very often your chance of getting a wrong answer rises, I guess, exponentially with the amount of time that sample is in transit, and the nature of that transit. And so it was just a question for our Zambian colleagues as to how often the samples they send away don't give a reasonable answer, or potentially give the wrong answer, even worse.
Actually,
Saha, can you answer that?
Sure. Us as UTH, we don't send direct outside, but just unfortunate, only the patients that they are able to afford the payment, these are the ones going to submit the sample to the private lab and it is sent. It is quite often they ask for another sample collection. We don't get a wrong answer, but most of the time they ask for recollection of sample. And this is how we advise the patient that they go the same day of shipment to minimize the days of shipment until it reaches to the referral lab. Because in Lusaka, there is nothing. There is no, or in Zambia, there is no flow cytometry. I was told that is one of the private labs, they are about to do the installation. They are in a training for 3 months. But also still it will not serve the low social people that they can't afford the payment, which we are really working hard to try to get something in UTH. We are in communication with Sysmex if they can put a machine in placement, but we haven't moved proper steps for that.
Thank you, Conor. Thank you for a very interesting talk.
Thanks, everyone.
Thank you, everyone.
Thanks, everyone, for showing up.